Hello students, this is the video for Lab 2. Bacteria reproduce asexually, which means that they divide in half and make two identical bacterial cells. However, they're still able to share genetic information. One way they do this is via small circular pieces of DNA called plasmids. Today you will be able to introduce a plasmid into bacteria that will give them new abilities. This image may look familiar to you from high school biology. Essentially, these are the different ways that organisms can reproduce. So, based on what I just said, think to yourself, which one am I talking about? Okay? I am talking about binary fission. Bacteria reproduce asexually, which means they just get their own DNA. They don't get to share DNA unless we mechanically add new DNA to the bacterial cell. Because of their simple reproductive processes, we use bacteria to carry special DNA into different organisms and assimilate that into their DNA. Now, looking at the other side of the paper here, we have mitosis and meiosis. So take a moment to think to yourself, what does this mean and which one do I do as a human being? Hopefully you know that human beings engage in sexual reproduction, and therefore there needs to be a way for the DNA from one individual and the DNA from another to co-join within the cell and then re-separate so that you don't double the amount of information inside of a single cell. And that is what we're seeing here on this right side, okay? So over here, the different colors are the two individuals involved in the sexual reproduction. But then look down here, we have chromosomal separation. So in the end, your sex cells, which are these gametes, okay, that one's the sperm right there with the tail, and this one is the egg. Notice it's much larger. There's much more investment put into a sex cell of a female, but that's not our topic today. The outcome is that you have the reduction of the chromosomes, the reduction of the DNA right back to what you started with, okay? But it's just so much simpler over here on the binary fission side of things, okay? Right here is the plasmid, this green thing, okay? That is replicated, so this one and this one are actually now copies. Identical copies. Not the same thing as over here because these copies are not identical, okay? Then you have chromosome segregation. So various things are happening in the cell to pull this away. And then some other things are happening to actually reclose the cell membrane. So we're thinking of these things as two separate cells. Oops, sorry. Okay, so what we're seeing in here is like the development of membrane around these contents. And eventually you have two whole cells. And now ask yourself, are these two resulting cells identical? Do they contain exactly the same DNA? They do. I dwell on this point for a few minutes only because this is kind of why we use bacteria in medicine as an organism to facilitate our research. It's very, very simple. Low stakes, not a lot of ethics with bacteria. Okay, so be thinking about all of these things when you're there in the lab. The simplicity of this process also drives what we're going to do in lab today, which is we're going to be able to give the bacteria new abilities just by our manipulations right here in lab. It's quite exciting. This is really what scientists do in the lab. This lab involves using micropipettes. So these things that are being held here are the pipettes. This is a way to pull up a volume, an exact volume of a liquid, and place it into another test tube. So before we talk about the procedures, let me just show you a few videos of us using the pipettes because it is definitely a skill that you need to acquire. So you just saw us holding the pipettes, and then your primary task is going to be to put a tip on the pipette. Never, 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 never put the pipette directly into a liquid because then it's basically ruined because you have dirtied it. It's dirty beyond repair. So these little boxes with clear lids are filled with tips. You never use a tip twice, you just use it and then you discard of it in the cup that's on your lab bench. And when you're not using the pipettes, you should put them into this stand. These things are pretty delicate, okay? And so there's a stand, there's plenty of places for different ones. The reasons why we use more than one is because they are appropriate for different volumes of liquid. But never just set it on the table. It's best to put it right back in the rack so it stays clean and doesn't get broken. All right, so here we have the pipette in the right hand and what is called the microcentrifuge tube. That tiny little tube is the microcentrifuge tube, okay? It contains 2 microliters of plasmid DNA and it has your initials on it. You're going to pipette 25 microliter alicate of Z competent cells from the microcentrifuge tube and add it to the microcentrifuge tube containing 2 microliters of plasmid DNA. These materials should be kept on ice, okay? Before you put them on ice, you're going to mix them gently by flicking the bottom of the tube. Let me take a quick minute to define some words that I used. So we already know what the microcentrifuge tube is, right? But we need to know what the plasmid DNA is and also what the Z competent cells are. So the plasmid DNA is the DNA of the virus. The competent cells are E. coli cells, and they're more likely to incorporate the foreign DNA if their cell walls are altered so that the DNA can pass through it more easily. So these cells are said to be competent, and we make them competent through a lab process that involves chemicals and temperature. You're going to incubate this on ice for 5 minutes. You're going to label the bottom of the 2LB agar plate supplemented with the 100 micrograms per liter of ampicillin. You're going to label that plate. Then you're going to add LB broth. We're going to apply all of this to the agar plate that we already talked about in the first video. You might be wondering at this point, what is LB broth? LB broth is the nutrition media for these organisms, microorganisms. It's basically Gatorade, okay? There's mention of this other kind of Gatorade called tryptone, but that's not quite as great as the LB broth in terms of feeding the microbes. Add 1 milliliter LB broth to the plasmid cell mixture that has been incubated for 5 minutes on ice. Pipette 10 microliters of this cell suspension on the appropriate labeled LB ampicillin agar plate. Sprinkle 5 to 10 sterile beads onto each plate and gently agitate the beads so that the entire surface of the plate is covered with the cell suspension. Avoid a circular motion that will concentrate the cell suspension on the outer edges of the plate. Pour the used glass beads into the container on the bench containing the Wescodine disinfectant. These will then be washed and re-sterilized. Do not reuse the beads. Your lab fellow will incubate the plates agar side up overnight.